MCQs, nucleases, restriction enzymes, DNA ligase and PCR. Which of the following best describes nucleases? A. Enzymes that synthesize DNA. B. Enzymes that break peptide bonds. C. Enzymes that cleave phosphodiester bonds in nucleic acids. D. Enzymes that synthesize RNA. Correct. C. Nucleases catalyze the hydrolysis of phosphodiester bonds in DNA or RNA molecules. Exonucleases differ from endonucleases in that they A. Act at specific internal sites. B. Cut at random internal points. C. Remove nucleotides from free ends. D. Add nucleotides to ends. Correct. C. Exonucleases digest nucleic acids from the 5 or 3 end, while endonucleases cut internally. The enzyme DNase is A RNA-specific exonuclease. B. Non-specific DNA endonuclease. C. Specific restriction enzyme. D. Ligase enzyme. Correct. B. DNase cleaves both single and double-stranded DNA non-specifically. RNase specifically cleaves RNA. A. At A, U bonds. B. After pyrimidine bases. C. After purine bases. D. At phosphodiester bonds adjacent to G and C. Correct. B. RNase hydrolyzes RNA after cytosine and uracil residues. Which statement about nucleases is true? A. They are used to join DNA strands. B. They degrade only proteins. C. They are essential for DNA proofreading and repair. D. They require light energy to function. Correct. C. Nucleases play a major role in removing damaged DNA segments during repair. Restriction endonucleases were first discovered in A. Viruses. B. Fungi. C. Bacteria. D. Algae. Correct. C. Discovered in bacteria as a defense mechanism against bacteriophage infection. The function of restriction endonucleases in bacteria is to A. Repair DNA. B. Degrade viral DNA. C. Replicate chromosomes. D. Join DNA fragments. Correct. B. They restrict foreign viral DNA by cutting it at specific sites. Which of the following is not a type of restriction endonuclease? A. Type I. B. Type 2. C. Type 3. D. Type 6. Correct. D. Only types I, 2, 3, 4, and B are recognized. Type 2 restriction enzymes are preferred in biotechnology because A. They cut DNA randomly. B. They require ATP. C. They cut at defined recognition sites. D. They methylate DNA. Correct. C. Type 2 enzymes recognize short palindromic sequences and cut at specific positions. Which of the following enzymes recognizes palindromic sequences? A. DNA polymerase. B. RNA polymerase. C. Restriction endonuclease. D. Ligase. Correct. C. Restriction enzymes recognize and bind to palindromic sequences in DNA. The recognition site for EcoRI is A. 5-GOT-C3. B. 5-IJ3. C. 5-GAP3. D. 5-TAG3. Correct. A. EcoRI recognizes GOT-C and produces sticky ends. Which restriction enzyme produces blunt ends? A. EcoRI. B. BAMHI. C. SMAI. D. HIDII. Correct. C. SMAI cuts symmetrically in the recognition sequence to form blunt ends. The palindromic nature of recognition sequences is important because A. It provides asymmetry. B. It allows both DNA strands to be cut symmetrically. C. It prevents enzyme binding. D. It causes random cleavage. Correct. B. Palindromic symmetry ensures both DNA strands are cut in equivalent positions. Restriction endonuclease EcoRI produces A. Blunt ends. B. Sticky ends. C. Circular DNA. D. Double breaks. Correct. B. EcoRI produces cohesive sticky ends with 5 overhangs. Restriction enzymes are named based on A. The DNA sequence they cut. B. The scientist who discovered them. C. The source organism. D. The type of DNA used. Correct. C. Named using the first letter of the genus, 2 letters of species, and strain/order. The main function of DNA ligase is to A. Synthesize DNA. B. Cut DNA. C. Join DNA fragments. D. Replicate RNA. Correct. C. DNA ligase forms phosphodiester bonds between DNA fragments. DNA ligase catalyzes the formation of A. Hydrogen bonds. B. Peptide bonds. C. Phosphodiester bonds. D. Glycosidic bonds. Correct. C. Ligase seals nicks in the DNA backbone by forming phosphodiester bonds. The cofactor required by T for DNA ligase is A. NAD. B. FAD. C. ATP. D. COA. Correct. C. T for DNA ligase requires ATP for joining DNA ends. DNA ligase from E. coli uses which cofactor? A. ATP. B. NAD. C. GTP. D. UTP. Correct. B. Bacterial DNA ligases generally use NAT as a cofactor. In Okazaki fragment joining, DNA ligase seals A30 and 5P ends, B50 and 30 ends, C50 and 5P ends. D. None. Correct. A. DNA ligase links the 30 of one nucleotide to the 5-phosphate of the next. Which of the following is not a function of DNA ligase? A. Joining DNA during replication. B. Joining DNA in repair. C. Cleaving phosphodiester bonds. D. Sealing recombinant DNA. Correct. C. Ligase joins, not cuts, DNA strands. PCR was invented by A. Frederick Sanger, B. Kary Mullis, C. Watson and Crick, D. Arthur Kornberg. Correct. B. Kary Mullis developed PCR in 1983. PCR amplifies A. Proteins, B. RNA directly, C. Specific DNA segments, D. Lipids. Correct. C. PCR selectively amplifies a specific DNA sequence exponentially. The heat-stable enzyme used in PCR is A. DNA polymerase I, B. TUC polymerase, C. RNA polymerase, D. Ligase. Correct. B. TUC polymerase from Thermus aquaticus resists high denaturation temperatures. Which step of PCR occurs first? A. Extension, B. Denaturation, C. Annealing, D. Cooling. Correct. B. Denaturation, 94 to 96 degrees Celsius, separates the DNA strands for primer binding. Annealing temperature depends mainly on A. Enzyme concentration, B. DNA template length, C. Primer melting temperature, D. Mg superscript 2 concentration. Correct. C. Annealing temperature is set a few degrees below primer Tm for specificity. The purpose of primers in PCR is to A. Degrade DNA, B. Initiate DNA synthesis, C. Cut DNA, D. Ligate fragments. Correct. B. Short oligonucleotides act as initiation sites for DNA synthesis. In PCR, exponential amplification occurs because A. Each cycle doubles DNA, B. New strands serve as templates, C. TUC polymerase is reused, D. Both A and B. Correct. D. Each cycle doubles target DNA, new strands become templates in subsequent cycles. Which of the following is not a PCR component? A. Primers, B. Template DNA, C. DNA ligase, D. Nucleotides. Correct. C. DNA ligase is not required, TUC polymerase performs synthesis. Which of these steps occurs at 72 degrees Celsius? A. Denaturation, B. Annealing, C. Extension, D. Termination. Correct. C. TUC polymerase extends DNA strands optimally at 72 degrees Celsius. Real-time PCR, qPCR, differs by A. Using fluorescent markers, B. Not using primers, C. Lacking TUC polymerase, D. Being non-quantitative. Correct. A. qPCR monitors amplification in real time using fluorescence. Reverse transcription PCR, RT-PCR, uses A. DNA as template, B. RNA as template, C. Proteins as template, D. None. Correct. B. RT-PCR converts RNA into cDNA using reverse transcriptase. PCR cycle count depends on A. Template length, B. Desired yield, C. Enzyme type, D. All of the above. Correct. D. All factors influence the number of amplification cycles. The purpose of Mg superscript 2 in PCR is to A. Stabilize primers, B. Act as enzyme cofactor, C. Prevent denaturation, D. Bind DNA ends. Correct. B. Mg superscript 2 is a cofactor for TUC polymerase activity. Which PCR variant allows amplification of multiple targets in one reaction? A. qPCR, B. RT-PCR, C. Multiplex PCR, D. Nested PCR. Correct. C. Multiplex PCR uses multiple primer sets to amplify different DNA sequences simultaneously. Nested PCR increases specificity by A. Lowering temperature, B. Using 2 sets of primers, C. Adding ligase, D. Reducing cycles. Correct. B. 2 primer pairs reduce nonspecific amplification. Hot start PCR improves A. Enzyme degradation, B. Nonspecific amplification, C. Primer dimer formation, D. Both B and C. Correct. D. It prevents premature amplification before denaturation. Touchdown PCR involves A. Gradually decreasing annealing temperature, B. Increasing cycle number, C. Changing polymerase mid-cycle, D. Extending reaction time. Correct. A. It begins with high annealing temperature to enhance specificity. The DNA yield doubles theoretically with every A. 2 cycles, B. 3 cycles. C. 1 cycle. D. 10 cycles. Correct. C. Each PCR cycle doubles the DNA copy number. Which of the following techniques uses PCR? A. DNA fingerprinting. B. Gene cloning. C. Disease diagnosis. D. All of the above. Correct. D. PCR is applied across forensic, clinical, and research biotechnology. The first thermostable polymerase discovered for PCR was from A. E. coli. B. Thermus aquaticus. C. Bacillus subtilis. D. Archaea. Correct. B. T. aquaticus yields the famous Tick polymerase. A typical PCR reaction runs for about A. 1 cycle. B. 30 to 40 cycles. C. 5 cycles. D. 100 cycles. Correct. B. Around 30 to 40 cycles are used for optimal amplification. PCR can detect mutations using A. Random primers. B. Allele-specific primers. C. RNA probes. D. Enzyme markers. Correct. B. Allele-specific PCR identifies base pair mismatches or mutations. PCR cannot amplify A. DNA template. B. Highly degraded DNA. C. cDNA. D. Short sequences. Correct. B. Severely degraded DNA lacks complete template for amplification. Which enzyme synthesizes cDNA for RT-PCR? A. Tick polymerase. B. Reverse transcriptase. C. DNA ligase. D. Restriction enzyme. Correct. B. Reverse transcriptase makes complementary DNA from RNA. Quantitative PCR determines A. DNA size. B. DNA quantity in real time. C. Sequence mutations. D. Protein concentration. Correct. B. qPCR quantifies amplified DNA during the reaction. Which fluorescent dye binds double-stranded DNA in qPCR? A. Ethadione bromide. B. SYBR green. C. Comacy blue. D. DAPI. Correct. B. SYBR green emits fluorescence upon binding dsDNA. PCR was awarded the Nobel Prize in A. 1983. B. 1993. C. 1975. D. 2000. Correct. B. Kary Mullis received the Nobel Prize in Chemistry, 1993. Which parameter primarily affects PCR specificity? A. Annealing temperature. B. Extension time. C. Denaturation rate. D. Buffer pH. Correct. A. Proper annealing temperature ensures primer specificity. The most critical step ensuring PCR fidelity is A. Template purity. B. Polymerase accuracy. C. Primer design. D. All of the above. Correct. D. All these factors collectively ensure precise DNA amplification.